So for this homework, you're going to be working on some slightly more difficult genetics problems. Let's start with this example. In saber-toothed cats, white teeth are dominant to black teeth, and solid-colored fur is dominant to stripes. We cross a black-toothed striped cat with a white-toothed solid cat, and all of their offspring are white-toothed and solid colors. So this leads us to a couple of questions that we're going to answer together. So our first question is, what are the genotypes of the mom and dad saber-toothed cat? So we know from the information before the question that we have one cat that's black-toothed and striped, one cat that is white-toothed and solid. So in this case, it doesn't really matter which is the mom or dad. We're not going to identify. We just know that these are the two parents. This is what it tells us. So we know that black-toothed and striped are both of the recessive traits. And so the genotype must be homozygous recessive. We use T for the teeth and S for the stripe or solid. So black-toothed and striped is going to be little t, little s, little s. Whereas white-toothed and solid, which are both of the dominant traits, give us two options. The big T, big S, little s, or big T, big S, big S. And we may be confused about which one of those it is, unless we look back at the question. So the question tells us that all of the children of these two cats are white-toothed and solid-colored. So that means that none of the children have this black-toothed stripe genotype here. And so in order for none of the children to have that, this is not a possible genotype for a white-toothed solid parent. So the two genotypes of our parents are this and this. One homozygous dominant, one homozygous recessive. So that leads us to our second question. What are the genotypes of the progeny? So we already know what the phenotype of the progeny is. We know from the question that all the progeny are white-toothed and solid. But this is asking for the genotypes. So if you need to, you can make a Punnett square. I'm not going to because this one's pretty simple. So we know that our parents here are big T, big S, and little t, little s. So if we cross those, there's only one option that we get, and that's big T, big S, little s. So this is the genotype of all of the progeny. And as you can see, like I said in the question, each of those are white-toothed and solid phenotype. So go ahead and pause the video, try to figure out what those four possible gametes are. 4 from the mom, 4 from the dad, and then we'll be back. So these are going to be the 4 possible genotypes. This is what you should have gotten. The 4 for each parent, what they can contribute. So the next step for you is going to be to make a Punnett square. So use these different gametes here and line them up like you normally do on a Punnett square. I'll give you a hint. This Punnett square is going to be twice as big as any Punnett square you've ever done. So take it, multiply it by 2. You're going to have 4 gametes on each side. So take a minute to try to do that on your own. This is probably the toughest part of the video and of your homework, but it's important that you're able to figure this out for the rest of the questions. So after the completion of your Punnett square, you should have noticed that these here on the left are all the possible genotypes that Punnett square gives you. And so I've also got the numbers next to them of how often they appear. And those numbers, that's what makes up our genotypic frequency. So essentially a genotypic frequency is how often related to the other genotypes does one genotype appear. So it should be written like this as our genotypic frequency. It looks a little bit prettier that way, but it's important to know what each of those numbers mean. So that's what we have here on the left. So now go ahead and try to do the same with the phenotypic frequency. So this time, instead of taking each genotype, take only the possible phenotypes. So, we've got our two options. So go ahead and try to figure out what the phenotypic frequency is from that. So you should have figured out that our phenotypic frequency is 9 to 3 to 3 to 1. So if you didn't get that, go ahead and do the same thing that we did here on the left, and this time do it with the phenotype so that we can figure it out, and then I'll pop those up here in a second. So when we break it down, this is how we see our phenotypic frequencies. So that 9 represents all the cats that are white teeth and solid coat colored. So that's any of the genotypes that have at least one big T and at least one big S. And then 3 black teeth solid coat, you can see 3 white teeth striped coat. It continues to go down. And so this is how we show our phenotypic frequency of a 9 to 3 to 3 to 1. So it's important to remember these two frequencies, the genotypic and phenotypic frequencies, because these will be exactly the same any time that you cross two heterozygous species that are heterozygous at both loci. So these are both heterozygous for the fur type and the teeth color. So if they're heterozygous for both, you always will see this frequency. All right, so the next question is, if I got a ratio of 1 black-toothed striped cat to 3 white-toothed solid-colored cats, what can we conclude about these genes? So I want you to take a minute to think about this. This is the normal phenotypic frequency that we should see. This is what we calculated in the last question. But for some reason, when we actually did the cross, we're seeing something a little bit different. For some reason, these two in the middle aren't showing up. So go ahead and take a minute to think about why that could be. So there's a number of different things you could have come up with as an answer for this. But what happens is it has to do with crossing over and the fact that these two genes are on the same chromosome. And so these two genes are actually very close together on the same chromosome, so when they cross over, they tend to cross over together. That's why we see a lack of these two in the middle, is because they're always jumping together. So we only see two different phenotypes. And so when this occurs, this is something to keep your eye out for, is when those two middle ones don't show up, almost every single time, it means that the genes are linked. And what linked means is, like I already mentioned, they're on the same chromosome very, very close together, and so they always cross over together. And so that's the answer to this last question of what we can conclude about these genes. It's that they're linked genes. So the way that we figure a problem like this out is by looking at the recombination frequencies they give us. So the higher the recombination frequency, the further apart two genes are on a chromosome. So we can see here to start, it gives us tail length and nostril size. And the distance between them is the furthest that's listed here. So let's just go ahead and start by putting those on the end. So this is the way that in your mind you should be thinking about a problem like this. So you say, well, from what I know, those are the furthest, so we're going to place them on the end. So then look at the next one. Tail length and tooth color are 25 apart. So we can drag tooth color on here and, I don't know, put it a little ways away from tail length. So then we see tooth color and fur pattern is 6 centimorgans away. So we've got tooth color already on there. So we can come in and we can put fur pattern there close to it. Then the next one that it gives us is tooth color and eye shape. So between tooth color and eye shape, it's 23 centimorgans. So we've got eye shape here. We'll go ahead and bring it up. And then we also can see that nostril size and fur pattern is 39. So once again, here's our fur pattern, and our nostril size is on the far right there. So we can go ahead and place them in order. This is a relatively easy one to do. You may have a bit harder one in the homework. But then you just place them up in order, and you can see, okay, tail length, nostril size furthest apart, and then go through each one and make sure that they look like they're in order. It doesn't matter the exact spacing on here, relative to where I put them. This just asks which order they come in. So now you can say that tail length is first, tooth color is next, then fur pattern, then eye shape, then nostril size. That's the order that these genes go in. So one common mistake that all of us make is, so we're getting back to adding a fur pattern, and we know that it's 6 centimorgans away from the tooth color. And so, why didn't we add it on this side? Some of you may have done that originally. I know it's a little bit messy now that we have all these numbers and stuff up, but why wasn't fur pattern between tooth color and tail length? And you might have put it there originally and said, well, these are 6 centimorgans apart, it still looks good. But then later on you would have come and seen the very end, oh, nostril size and fur pattern is 39 centimorgans. Well, if it's there, you can add up the ones in between, the 6, the 23, and then take the 70 and stuff, and you're like, whoa, this isn't quite making sense here. So that's how we figure out, oh, okay, this fur pattern is on this side. And so in the one in your homework, there'll be more tricky ones like that, where you're unsure of which side to put it on, but then later you're able to find out because of some of the other numbers. So always watch for that as you're doing problems like this.